cell behavior is influenced if a molecule binds to a receptor but doesn't activate the receptor, right? So like if I have a cell that is a cancerous cell and it's using like an RTK to stimulate cell growth, if I have a drug that binds to RTK but doesn't activate it and prevents the RTKs from clicking together, I mean, that's the mechanism of an antagonist. It sits in the receptor pocket and prevents the ligand from accessing it. Caffeine was an example of that. I don't need you to know the specifics of caffeine, but you need to know what an antagonist is. And you also need to know what an agonist is. So like, if I expose the cell to a drug and I see that that drug stimulates the signal pathway more strongly than the natural ligand, then that drug is not an antagonist, it's an agonist. So if this is the drug and it binds to the receptor, and suddenly I see that the signal pathway is on very strongly, that would be an agonist as opposed to an antagonist. So that's it, as far as drugs go. The drug, you know, if I'm talking about a drug could also go into the cell and, you know, if the drug inhibits AC, or if the drug inhibits the G protein's ability to let go of GDP, you know, I can ask you to predict the effects of that. But you don't have to memorize any specific drugs. It'll be given to you in the form of a figure, and you've got to interpret the figure and figure out what the drug is doing, how the cell's response will change in the presence of the drug, and why. What else? Yeah.
Can you go through the RTK signaling pathway?
Yeah, so an example of the RTK signal pathway is insulin. So here's an insulin receptor, another insulin receptor. When insulin is present, it will bind to an RTK. It must bind to another RTK, and that will allow the RTKs to dimerize or click together. In their dimerized form, they can phosphorylate each other. These phosphate groups serve as attachment sites. It's typically multiple phosphate groups, but I'll just draw two for the sake of clarity to simplify it. Once these RTKs become phosphorylated on the cytoplasmic side, proteins can attach to those phosphate groups. They sort of serve as docking sites. And these proteins can now change shape and become active. So this protein might trigger a kinase cascade. Maybe it phosphorylates pKa and triggers a kinase cascade. This protein might activate another protein that causes IP3 levels to ultimately go up, and therefore calcium levels to rise. So you don't have to worry about what specifically is happening here. If you know what is happening in the G protein pathway, essentially similar things are happening here. So it's not like you have to remember what the G protein transduction pathway looks like versus the RTK pathway. The thing that is going to be emphasized for any RTK question is the fact that the receptors have to click together and phosphorylate each other. Does that make sense?
When they phosphorylate, the protein's just attached to the phosphate groups.
Yes, and it's not like the proteins are grabbing the phosphates off. The phosphorylated sites are like attachment sites for these proteins to click onto and change shape, and then they can become active in this new shape as they're attached to the RTKs. They literally stick onto the RTKs, change shape, and then while they're stuck onto them, they help with the signal transduction process. Yeah.
Is there like a specific grouping or name for the process that RTK uses, like that type of receptor that like a name? You need to know that.
You don't need to know that. Yeah, I mean, yes, it's the Roth-MEK-ERK pathway, but you don't need to know that. But like, is there a classic RTK pathway like the G protein pathway? Yeah, it's this, but it's beyond the scope of this class, so you don't have to worry about it. You might, but yeah, let me just leave it at that, because I don't want to confuse you. What else? I mean, since no one's saying much, the thing I'll mention here is that, remember, for topic 4.4, topic 4.4 is talking about feedback. So like, if this is the insulin pathway, you might get a figure like this, and then it might transition into a question about feedback. So remember, positive and negative feedback. Negative feedback is just any movement away from a narrow range is countered to bring the system back into that narrow range of conditions. And so in the case of insulin, it's the rise in blood sugar that triggers the release of insulin that will bind to liver and muscle cells. The liver and muscle cells, in response to insulin, will insert transport proteins on their membrane so that glucose can exit the blood and diffuse into the liver and muscle cells, bringing the system back to that ideal range. An example of positive feedback would be like, let's say, ethylene is a common example. So let's say this is a cell that produces ethylene. That ethylene will diffuse out of the cell and into other cells by simple diffusion. And these cells respond to ethylene by producing more ethylene. And so it's this positive feedback loop because the presence of ethylene triggers the creation of ethylene, which triggers the creation of more ethylene. So like if you look at this system where you have ethylene concentration, as soon as those ethylene levels rise, rather than ethylene production being cut off, which would be a negative feedback example, ethylene production accelerates and kind of runs rampant. But for any positive or negative feedback example, I could say, okay, here's the mechanism. Now let's zoom into the cell and let's talk about the signal transduction response aspect of it.